very important, but I thought we would zoom out a little bit and talk about DNA repair in general, if that's okay with everybody, and try to pick themes. I would like to ask for your help with discussion, if you don't mind, in part because I don't like to talk the whole time, and in part because I think I would like to relay more concepts than specifics. So, just by way of a question, you guys take something about DNA repair and MPG in the first class, right? In a little bit, right? So I'm going to try to ask you to recall some of that knowledge, and feel free to pull the reviews that I had shared with Cody or Google, and we can try to find our way through this. Does that seem okay? So maybe the first thing we can talk about, and this is sort of my introduction, why is DNA sequence fidelity important? It's kind of a simple question, but just to review.
The basis for proteins and structure. It's the basis for structure and protein. It's like the blueprint.
So if you lose the blueprint, what happens?
Anything.
Yes, that's the right answer, but give me an example of what anything is.
Loss of function of a protein from deletions in base pairs of a gene.
But if you think about evolution just a little bit, like why does evolution have to create mechanisms to maintain the fidelity of a genome? I don't want to pick on people. But
well, I think it's important to have some consistency, like just on the basis of like being able to have a species or like a grouping of organisms.
Yes. In order for a species to survive, it has to maintain its genome somehow. And what's the counterpoint to that? Why is it helpful to have imperfect DNA repair? So the fidelity, let's call it, for lack of a better word, a dominance in the kingdom of life.
It allows a bit of wiggle room for errors that could potentially be useful.
Yes, it should ideally be, imperfect in order to allow possibility, right? Let's call it possibility. And when do those possibilities come in handy? And if you think of Darwin's theories and
Like extreme environmental changes.
extreme changes, new changes, like sort of like what's happening in our environment right now politically, new ideas, something new. So it can't be perfect, otherwise it would be so rigid it wouldn't allow for evolution to happen. I think those are the two things I wanted to point out. So, just what are the threats to the genome then, if we had to categorize them? Or
I'm going to share screen right quick.
oh yes, thank you. I think one of them is what we just talked about, like threats, environmental threats. Can you think of things in our environment or that you might know about or you might have read about it in those reviews? One of them was really, really long, so if you want to cheat, just look at the tables. And that's the Elledge review. What are some sources of environmental threat to the genome?
Like
UV light.
UV
or, like,
light,
smoking, things like that.
And do you remember, Lori, what the damage is there?
Do you mean dimer?
Sorry?
Do you mean dimer?
What's the dimer?
Timing?
Diamond, yeah,
Diamond.
yeah. What else?
Oxidation.
Yep. So let's divide them into like endogenous and, so this is, you know, reactive oxygen species. Other things that cells make byproducts of that damage the DNA. Let's name one more exogenous.
Insulating.
Radiation, yep. So ionizing radiation. And how does that damage the DNA? Does anyone know? So it creates double strand breaks. And these are two fundamental pathways that I'd like to talk about and think about with you today. So, and I also, you know, even though we are a human genetics program and I'm only trained as a medical doctor, I want to help us think of DNA repair sort of as a conserved mechanism across evolution. So I know we talked about extreme temperatures, so some microbiota, they live in volcanoes, and they have adapted to live and survive in temperatures that exceed the denaturing temperature of DNA. And I have spent the weekend trying to read about that because I think it's very interesting compared to the human syndromes. But the human syndromes are fantastic, and I hope you'll appreciate. But it is remarkable. And what does anyone know? I don't know who said UV light. I think it was Zoe. How far in evolution does mechanisms to repair these thymidine dimers exist? Or let's say, who of your favorite species has to have that too?
Yes, most of them.
Most of them,
Yeah.
exactly. So even bacteria, in order to live above ground, have to have a DNA repair mechanism that fixes UV light. Otherwise, it would threaten the survival of bacteria. So I think those are the things I wanted to just point out, and thank you for your help with that. So I think here are some of the tables in that review. These are endogenous things that happen that have mechanisms. We talked about oxidation, etc. And the mechanism for telomere shortening, we might as well get that out of the way now. What's the endogenous repair problem with telomeres, from what you remember from MDG class? Something related to the ends. Different page in that same lesson. Different slide. Remember the end replication problem, that the telomeres cannot, the ends of chromosomes can't be fully replicated, so there's always a possibility of shortening, and unless there is something that elongates that, like this cool enzyme called telomerase, you can't overcome this. And of course, if you look in the human population, this has been known for a long time, you have telomeres shorten over time as we age, right? This is a program that is created, and we're going to talk about the consequences of that. So this is not sort of a damage like these kinds of damage, but it is a program process that keeps track of time in cells, at least in the human and mammalian context. So someone said ionizing radiation. Sorry, yeah, thank you. This is very striking, and it's so this table quantifies the amount of radiation in day-to-day life activities, And ionizing radiation creates a very specific kind of damage, which is a double strand break, which we can talk about why that's important specifically. And it talks about the number of lesions per cell. So I don't know if anybody has seen anything like this, but basically, just for context, getting a chest x-ray is almost 20 times, sorry, if you travel, where's the airline travel? If you travel on 20 trips, it's basically the equivalent of getting one chest x-ray. And if someone says, hey, I'm worried about your family member, let's get a CAT scan. Please remember this slide at the time, because if they are just doing that because the patient is asking, which is what happens in the clinic where I'm going later today, too many CAT scans, that is a lifelong increased risk for cancer. It has to outweigh the risk has to outweigh the benefit here. And that's basically 200 times more than a chest x-ray. That's a lot, right? So, for example, some people argue that mammography to screen for breast cancer is harmful or it doesn't save lives because you might detect more breast cancers at an earlier stage, but you're actually exposing the patient to radiation, and who knows what the effect of that is. And you might actually potentially be causing the breast. So you get the idea that unless, and then of course, what did the people in Hiroshima die from, if you remember? Or what would you guess they might have died from?
You already got the lecture from Bert.
Sorry?
You
Oh,
already
the
got
lecture
it.
from Bert, okay.
So,
Did he
yeah.
talk about these things too?
No,
Okay.
but he did talk about how many mutations we need for certain cancer and other cancers.
Yes, but those patients, they did not die immediately from cancer, right? Because in order for this to happen, this is the number of lesions per cell, and you assume that there's some repair capacity, right? We're going to talk about those repair mechanisms. But the first thing that will happen is the cells that turn over the most are going to die. So these patients, a lot of them died immediately from blood problems, from stem cell failure in the blood, because the blood turns over so quickly. So
granulocytes.
Exactly. So the first, let's say if this is years, the immediate problem is hematopoietic stem cell failure, and that's like within the first 22 months. And then the cancers start to arise here.
When you do a bone marrow transplants in mice, you irradiate
Exactly.
to kill all the stem cells, and then you can give like a whole bone marrow from another mouse that has a little bit of everything, but including granulocytes. But if people that study say they want to find out what's the hematopoietic multiprotein, the real one cell that describes everything, they would sort a couple of these, a couple of that, and put it in. But that takes a long time to give rise to this. So you have to do like some other stuff to aid it survive that period of time until the stuff comes through.
Exactly.
And granulocytes are the one that the last half-life is the shortest. Leukocytes last a little longer.
That's great.
They also die in
You
space
saw my
really fast.
slides already. But and one of those cancers, like Elena is saying, is leukemia. So the damage in stem cells, the point here, stem cells keep track of time. If the damage is too great, it will overpower their repair capacity. And the most immediate consequence of too much radiation, if it's a blast, is death. The cells die, the species die, etc. And then cancer comes later in multicellular organisms. And I'm going to apologize again. My talk is going to be human-centric. But please, if you'd like, and I found a lot of papers about DNA repair and archaea as I was reading this weekend, and I would love to share them with you because these are highly conserved mechanisms. that we borrowed from more primitive organisms or more interesting unicellular organisms is probably the better, less judgmental way than calling them primitive. So, okay. So, and this is important as to which, precisely for the reasons that Elena mentioned, what tissues are injured or receive the radiation is going to matter as to how they repair it. So hang on to this idea of cell turnover and immediate versus late consequence. Okay? So, how are we doing so far? Okay. So let's think of what would you have to do to repair a DNA lesion. And feel free again to look at those papers. Like, let's say you have so UV light comes in and creates these aberrant bonds between two thymidines. What kind of enzymatic reactions would you have to fix that if you're a cell trying to protect its genome and therefore trying to protect the species? No. So we can think through it together. You have to recognize it, right? So you have to have some machine to recognize the lesion. You have to have some endonuclease to excise the lesion. You have to have some kind of polymerase to fill in the lesion, right? So let me see. And for things like homologous recombination and homologous end joining, what other kinds of enzymes would you like to add to our list here?
One that acts on the back though?
A recombinase, maybe?
Yeah.
Yeah, great. So let's see what we got. So you have to recognize we got that excision, like you have to ligate it. Sometimes you need helicases to unwind that part of the region. And then you need the DNA replication machinery to fill that in. That makes sense, right? So all the DNA repair components that are very confusing to me even include these kinds of components. And I think, what I would like to relate to you is that probably it's not clear whether evolution created these multiple times, but probably they all came from bacteria having a recognition mechanism to repair UV damage. So the first classical pathway that I wanted to point out is this nucleotide excision repair, which is the canonical pathway historically. And you see all these combinations here of the steps that we basically outlined in my very primitive figure. But what I want to show you is, and talk about with you, is these things called XP. So there's XPD, XPA, XPF. Does anyone know what these things are? Sorry, it's hard to see here. But
You should have the review, no?
if you have the review with you, this is the Grant Stewart review. You see these components are called XPD, and they basically, replicate the nice list that we made here. Does anyone know what XP stands for in this context?
Zero.
Perfect, okay. And what is it? Tell me anything you remember about that.
They have, I want to say that it's specifically issues with UV radiation, thymidine dyes.
Perfect. So these are humans who are born with they're born normal, but as they are exposed to light, they get spots on their skin, pigmented spots. They get skin cancer, not surprisingly. And the way that these components, like I think if you were to meet an XP patient now and you're on an island and you graduated from our program and they say, hey, you're a human genetics student, human genetics and genomics student. Please help me find the mutant gene in this patient with xeroderma pigmentosa. What's the first thing you would do?
I mean, I would sequence them.
You would sequence, exactly, but how long have we known about the human genome?
I mean, not the actual sequence, not very long.
Not very long. 30 years? 30? 25 years? Somewhere between.
In the 90s.
Sorry?
Yesterday in the 90s when I was in.
Yes, yeah, in the 90s, right? So not so long ago. This entity of xeroderma pigmentosa was identified in 1960. So what people then were clever. They had no sequencing technology. There was no PCR. What they did was they would take cells from the patient. Can you see her? They would take cells from the patient. They would immortalize them, or let's say they would take fibroblasts, and they would expose them to UV light. And if the cells died, they made the diagnosis of XP. That's pretty interesting, right? So then how do you think they identified these components? They took the genome, a bunch of DNA, they chopped it up, and they tried to put pieces of it to see what would rescue that. Does anyone know what that word is? What kind of experiment this is?
Genesis?
Close.
Cloning.
Selective.
Sorry?
Cloning.
Cloning and complementation, right? So basically, the reason that these genes have such strange names like SPA, B, C, D, E is because they kept seeing patients and they discovered one gene. They had no idea what these genes did, but they just put them back and they knew they rescued the defect. And they would say, this patient has complementation group A, XP. Does that make sense? So this is essential for this, the sort of the historic context behind DNA repair mechanisms, because the patient cells became a way of describing these pathways and understanding these pathways. And these genes did not come from looking at homology in PyMol or some AI-directed things. It came from studies of patients who had a uniform phenotype, someone who discovered the connection between UV light, and someone who discovered all the components that way. And then some scientists later came up and they figured out, oh, this is an ligase, this is a recombinase, does that make sense? So, so what about this one? What about the double strand break? Why is this one of note? I'll give you a second while I erase yours. How are we doing, Kirby? Okay. Checking. Oh, nice flipped one more. Okay. So, take your time. I'm just. So the problem with, like, you saw the single-strand DNA. If it can excise, there's another template, right? But when you break the DNA in two, there's no parental strand that you can copy from, right? So you can. what's the only way you can repair this? Or what are the possible ways you can repair a DNA double-strand break that occurred when I was going to Europe last month? Oh no, I should have taken a vote.
You can smash the ends back together.
How are you going to find an end? You might, in desperation, if you're a double-strand break in a genome trying to survive, just find a random double-strand end and create a fusion. So that's probably how fusions happen, and that can be bad. It can be okay, but it can be bad. What if the cell is dividing? So I think let's try to reason through this ourselves. So if you have a double strand break and you are in SG2 and you have 2 copies of the genome, is that going to be different from whether you're in G1 or G0? Where you have 1 copy of the genome. It seems like you agree there could be different strategies to repair that break.
I'm guessing in the SG2 you could use the other one to
Yeah.
kind of fix.
Perfect.
The
Yeah,
repair.
that's the homologous recombination. That's perfect. That's going to be safe, right? You have another copy. That's high fidelity, serves the purpose of fidelity. But what if you're if you're in this phase of the cell cycle, you're either gonna maybe create a fusion. It's gonna be error prone, right? Let's just, we don't have to go through all the possibilities, but we agree it's gonna be error prone because the cell is gonna make something up to fix it. Now it turns out that the human mammalian system uses this error prone to create diversity in a certain compartment. Does anyone remember what that is? Thank you so much for your participation, by the way. I really appreciate that. What cells use this error prone to create multiple genomes that they in turn use to help the organism?
Like antibodies?
Perfect. Yeah. So this is exactly what we have VDJ4, right? B cells and T cells. They randomly reshuffle their genomes in order to create this error proneness. And in creating such a diversity, they can recognize seemingly unlimited amounts of antigens, either by secreting antibodies or by creating T cells that. Right, our T cell, it's remarkable. The immune system creates a diversity before it even sees the antigen. And then it expands. So that's great. So, okay, so now we are experts in DNA repair. This is the recognition machinery. This is going to signal, oh my gosh, there's a double strand break, please help me repair. And cells are either going to die, repair, and if the cells are not cycling, they're either going to keep the damage or they're going to repair it erroneously, or they're going to degrade, smiley face, repair from the other homolog. Perfect. Okay. So what do you think then is the most intuitive phenotype of humans that have mutations in these components here? Just following from what we just said about
Like
immune deficiency. Perfect. So these people are going to get immune deficiency if they can't repair, and they get cancer, and I'll tell you they get other things, but. This I will come back to, but this is very intuitive. So far it all makes sense, right? So So then, let's go to not sure, maybe the battery down or not sure. So, has anyone heard of this Fanconi anemia pathway? So I just want to mention this for the sake of completion. So Fanconi anemia is a disease just like XP, but these patients, instead of, we said XP is a sensitivity to UV light, right? These patients are sensitive to a kind of drug called mitomycin. So you don't really have to know that, but it comes in very handy. Because if you take the cells, where did we give them? Here. If you take an FA patient and you take their cells and you expose them to this mitomycin, all the cells will die. And this is very important because mitomycin is also a chemotherapeutic drug, and if you meet these patients and you don't recognize they have Fanconi anemia, you give them mitomycin, they also die. And now you know the trick of these historic ancestors of ours who had no human genome or IGV to look at or any sequence. They would do the complementation groups. And guess how many complementation, so the lesion though, I should tell you here, thymidine adducts in XP. Here in Fanconi anemia, what happens to the DNA, this is Watson and this is the Crick strand, these chemotherapy drugs create covalent bonds between the two strands. So that's a bit annoying because if you're the DNA replication machinery, you go through and you get stuck, right? So how do you excise that? This mechanism of repairing intercross-linked repair is more complicated than all the others because you have to break this, you have to break this. You get the idea, right? You can look it up. There's ligase steps, there's recombinase steps. So how many complementation groups are there? They're now to Fanc W. I think there may be even more. Like they're running out of the alphabet here, whereas XP only goes to 8, I think. So this pathway is very important because parts of repair of this cross-linking defect in Fanconi anemia is homologous recombination. There comes a point in the story where you break the DNA and cells that are cycling can invade the other strand, and you need the homologous recombination machinery, okay? So this pathway, people thought it was obscure. People thought that these conditions are rare. You know, some people like me were interested in these conditions. They cause blood disorders. And then they discovered, oh my gosh, parts of this pathway are mutated in adults who have cancer. This one should say BRCA2. So components of this pathway are mutant and predisposed to breast cancer. Everybody here has heard of BRCA1, right? And 2. And so that means that the inability to repair homologous via the homologous recombination pathway is cancer predisposing. All of these disorders are cancer predisposing. But I want to take a second now to talk about the zygosity issues of these disorders. So all these disorders, we talked about XP, we talked about, the double-strand break disorders, and let's call them immune deficiency for lack of, so I don't overwhelm you with names. And then we have Fanconi anemia. There's others, but what is the mode of inheritance of these disorders?
Autosomal dominant, I think I read.
Of double check, because these are rare and obscure disorders. Most rare and obscure disorders tend to be recessive, right? So why do you think, so that means if you're born, let's say, with FANC A, if you're a FANC A heterozygous human, there's probably no defect in the ability to respond to mitomycin. But let's say you're the parent of a child who has XP, the parent can go out in the sun, but the child can't. So what is it about the dosage requirement that this, like what percent of human genes are sensitive to half the amount, to this haploinsufficiency phenotype? Do you have a sense for that from your classes so far? We don't really know because we've studied these disorders that are recessive for so long that we don't really know, but maybe 10%. Maybe Kirby or Elena have a better number, but why is it that the DNA repair disorders are almost all recessive? Let's agree on that. Why would you think they might be?
I would think that they're recessive because, I mean, if you can't repair your DNA, you're probably either not being born or you're dying before you have the chance to reproduce.
That's right, and that keeps things at a perfect balance because we talked in the beginning, you need a little bit of permissive imperfect repair, so probably these heterozygous humans, maybe they have a tiny bit of a repair defect enough to create variation, but most of these individuals who have Fanconi anemia or XP, they're not fertile. They're often infertile, they can't have children, they often die in early life. So, and also, as we were just saying, it can't be that fragile. I think that's what I'm saying. Like the integrity of the genome cannot be liable to one copy being mutated, right? Because you see how many copies there are. We've just talked about like 60, 70 genes, and there's so much human variation. It's possible that any of us can have a heterozygous mutation in any of these things, right? So it's a safeguard, basically, to require that you lose 2 copies in order to maintain this integrity. So then, before we switch to the telomere, what do you make then of this BRCA finding that the components of the pathway are also mutated in adults with BRCA? What is the mode of inheritance in, like, if you know anybody or with a BRCA mutation, or Angelina Jolie, or any of the famous people who've tried to educate the public about these things? What's the mode of inheritance there? Is it recessive? Sorry?
I think it's dominant.
It's dominant, right? So here, you know, a classic pedigree would be like this, right? It's not I mean, many things can cause this, especially on the whole exam. But this is the BRCA 1 pedigree, right? But in the Fanconi anemia, it's often a founder mutation, which looks like this, so it's going to be like this. So let's say someone has a BRCA1 minus, and this is the BRCA. This patient is going to be a child. They're going to be very sick. They're going to have very strange neurologic defects, hematopoietic defects, etc. But this is going to be an adult who comes to my clinic and says, what can I do to prevent cancer? So how do how do you reconcile the fact that DNA repair disorders in general are recessive, but here we have this context? What happens in the tumor here? Looks like you're onto something, or I don't, if you're not, it's okay. No?
There's a loss of heterozygosity.
Exactly. So the tumor here is gonna be just like here. That's exactly right. So the tumor, this is the germ line, but the tumor is gonna be BRCA1. Okay. That's pretty amazing, right? The entire human is okay, but in the breast tissue or in the ovarian tissue or wherever, this person is going to lose the second copy and essentially be functionally liable to defective homologous recombination. Yeah. Okay. I thank you for putting up with all that. That was very good. I'm glad. Let's wait for questions. Yeah.
I was going to ask just quickly about BRCA1. Would it be considered a tumor suppressor gene?
Yes.
Okay.
It's tumor suppressor, and this is the second hit, the so-called second hit, right? I mean, you
Berg
talked
was-
about that with birds,
Yeah,
right?
Berg was saying something about a lot of times papers will show like there's a motive in here and it's similar to this, but they'll falsely call something a tumor suppressor gene even though it seems like it is. So that's why I was asking about that.
Yeah, this is we have many examples of this in the literature, but there are a few examples where there's a disease that's caused by heterozygosity and a disease by homozygosity. What other thoughts do you have so far before we keep going? Does it make sense so far, or? No? Does it work? Okay, thank you. So that's basically what we've been talking about, multiple types of DNA repair pathways that have evolved. And almost all of them are autosomal recessive. This one, the mode of inheritance, is dominant, but in the tumor it's recessive because of loss of heterozygosity, as was so articulated. So I just want you to remember this as you think about people claiming a cancer predisposition gene of DNA repair. It will likely be in this category, except the telomere. The telomere is an exception, and that's what we're going to talk about for the next few minutes. So as you might know, I have taken interest in these telomere disorders, and in aggregate, it doesn't matter, the fact that they are very common doesn't make them more important, but I think they illustrate points related to what we were just talking about. So as we mentioned, the end replication problem here sets it up so telomere length shortens as we age. We are programmed to shorten our telomeres with age. That is invariable. That is, and some people are born with short telomeres, and they have mutations in telomerase, and the catalytic component of telomerase is this TERT. Now, the point I want to make here is that 99% of these disorders are autosomal dominant. Okay, so there's no second hit required. These are not tumor suppressors. And one can argue that all humans, if they shorten their telomeres with age, have a DNA repair deficiency, right? Because we all shorten our telomeres. And one can argue that this is just an exaggerated process of normal aging. How interesting. We are already, even when we have wild type levels of telomerase. So let's think, how many telomere ends are there in non-cycling cell. 46 times 292, right? So 92 telomeres. And we have this we have telomerase, which is pictured here. How many molecules of telomerase do we have per average cell? It's like 6 to 10. Oh my gosh, like it's programmed to prevent elongation at every telomere. It's almost like made that way. And telomerase is expressed very, very transiently during development. So the telomere length that we are born with basically is decided around the blastula phase. And it's downhill, quote unquote, from there. What?
I always say, everything is downhill from the
There is a positive end to the story. but for those of you who are going to be there at the Journal Club on Friday, the discussion there is how cancer cells overcome this downhill phenomenon in order to stay immortal, right? So we talked about this. I'm going to skip here, but just like the Hiroshima exposure, the most common, or let me rephrase, the most obvious phenotype of short telomere length is that the bone marrow falls apart, sort of like what Elena mentioned. So, where did the graph go? After Hiroshima, in the immediate term, stem cells died. People died from bone marrow failure. In short telomere disorders, if you are born with short telomeres, this is the first phenotype that occurs in childhood too. But here, it's not an exogenous double-strand break. It's an endogenous failure to have the replicative potential. Because I should tell you that here we talked about double-strand breaks. The telomere is also a double-strand break of sorts. Right? And people have studied for a long time, why isn't it that this end just fuses randomly to another chromosome? And that would be also disastrous for mitosis and segregation properly and all these things. But it just happens that the telomere has protective mechanisms that prevent it from fusing to the other chromosome. So, and these two pathways signal to the same p53 mechanism. So the cells synapse, cells die, etc. How are we doing? Is this... Take a deep breath, yeah. How would you repair this defect? Elena gave a clue. If you want to cure someone from their bone marrow problems related to short telomeres. Or stem cells from someone else. It works. And that also proves that it's a stem cell disorder. The other phenotypes that happen in these patients and mice, this is from the mouse actually, and is the gut. These are villi and intestines, and that's also another high turnover tissue, and these patients have malabsorption, they're often thin, etc. So, part of the reason I have been interested in these disorders is I met this family a long time ago, and they had features that looked like the DNA repair disorders, but didn't have mutations. So their bone marrow fell apart. This guy, he was a college student. He was 22 at the time. He went gray, etc. He had immune problems. He had immunoglobulin problems. But the clue here was that in his family, things became earlier and more severe with each generation. So what do we call that? You've taken that in one of your classes. A more severe onset with every generation. A genetic. Perfect. And genetic anticipation had been identified, I'll go back here, in mice before in the humans. So remember what we talked about here. This is a different paradigm. In XP, in Fanconi anemia, in ataxia telangiectasia, all these disorders, the human came first. And then we learned about the biology. Here, telomerase was discovered in a primitive organism, Tetrahymena. And then they made the mouse. And then I recognized this family, so it's a backward story. I'll tell you why the human studies are important. But when I was in the Greider lab, and Carol Greider used to be a faculty member in our program, they had a colony where they bred head-to-head telomerase RNA knockout mice. And they kept track of the genotype. And this is very striking because this is the survival curve of the knockouts from every generation. So the wild types lived this many days, and then the G1 and the G2 and the G3, etc. And then the colony disappeared, it vanished. How does this graph strike you? Does it strike you? Maybe it doesn't strike you, I shouldn't assume.
It's kind of scary, like within a couple of generation times, like
Yeah.
it's done.
Yeah.
Also sad for the person who made the line.
Made the what?
Sad for the person who made the house.
Because they
Lost
died.
it.
Well, this was like 3-4 years of keeping track of survival. But I think to me, well, it looked like the human family that I showed, right? So the genetic anticipation gave a clue. And also it shows that the telomere length is a powerful predictor of life or death decisions on an organism level, not just in cell culture. So the other thing in this family that was obvious to me was that in this genetic anticipation happened also for lung disease. And the lung disease was called idiopathic pulmonary fibrosis. So these are CAT scans from the same person who was one of the early people in our study. And you see that there's more scarring in the lung. Now, we haven't talked about anything related to the lung so far. We talked about the immune system, we talked about the blood. And it turned out that mutations in telomerase in that the family that I talked about had a mutation in TERT. So you can see that if we are all haploinsufficient, let's say we are all insufficient for telomerase, and these people are born a little bit shorter, and they shorten at a faster rate, they're going to reach the threshold sooner. So based on the observation in that one family, we ended up identifying mutations in telomerase in people with this lung disease very commonly. And unlike the young person in that family, look how old these people are. They're aged. Their lung is aged. In fact, if you look at people over the age of 70, and we do CAT scans on all of them, don't do it, it's too much radiation exposure. We all acquire this kind of hair graying in the lung. This is a premature aging phenotype in the lung. And I want to use that to talk about the biology of DNA repair. So at the time, we had those mice that I talked about that were null for telomerase, and no one had studied their lung because no one thought that it was worthwhile to study their lungs. And when you look at if these mice are exposed to room air and they're just sitting around, they have no defect in their lung. You see there's nice alveoli and everything, but And we did this experiment over and over again without luck, and I thought it was the end of my livelihood. But then someone told me to expose them to an injury. And the injury here was cigarette smoke. So there was a chamber in the School of Public Health at the time, I think it's still there, and you could put the mice there and expose them to cigarette smoke, or I guess in today's language you could say vaping. And we then looked and they developed the phenotype, but this is the slide that I want to show you. So after 6 months of exposure, you can count the number of double-strand breaks in the lung after smoking. So in the short telomere, there's double-strand breaks, but they're at the telomere. After smoking, you increase the number of of double-strand breaks. But here, it's cumulative. You see that? It's cumulative. And this damage is not at the telomere per se, it's random in the genome. So why do you think then people see this destruction of the air spaces? Like, how would you reason based on that DNA damage accumulation? What do you think is happening to those cells that are accumulating more and more?
They can't repair, they might just be looking at
They can't repair, they either are dying or senescent. So senescence is basically a state of lack of replication. I can't move on through the cell cycle. So this made sense, right? It made sense that, but in particular, it made sense because but it was also paradoxical, and I'm sorry I'm dragging you into all the paradoxes, but you're very smart, and I think you're following me. In the gastrointestinal tract, no? What? Were you going to say something? Okay. In the GI tract, the entire epithelium turns over in 7 days. You label this with some nucleotide analog, and it labels. But in the lung, in the same period of 2 weeks, there's hardly any turnover. So why do you think there is no repair in the slow turnover tissue? So where are we here? So no repair, or like how do you relate that to this diagram here of homologous recombination here, non-homologous end joining?
Maybe the slower turnover cells just because they're going initially are already kind of slower overturning, they're not going through the cell cycle enough times or through enough period of time to have DNA repair, DNA damage recognition software to like recognize that there's a problem. So it's like the problem if the problem occurs early on, it's going to continue to occur.
That's right. Thank you so much. That was perfectly said. And that's why people who smoke for 10 years, even if they stop smoking, their risk of lung cancer, is lower a little bit, but it's a lifetime risk. Because once you damage a slow turnover tissue, the repair capacity is diminished, and the fidelity is diminished. So when you're born with short telomeres, with a certain kind of damage, and you add to it another damage, you reach a threshold. So I think this gives a sense for exactly what we were talking about, that the signal, like if you look at a cartoon in a review article, it might say double strand break, DNA damage signal, p53 phosphorylation, cell death. It's going to say that. But the fact is that there is a threshold effect. It's not just one, it's multiple things that have to happen to the genome in terms of double strand breaks. And cumulatively, you can see it as a defect like this, an aging defect. This is called emphysema. It turns out that there's profound sex differences, and females are more likely to get this for reasons we don't understand. The DNA repair potential is different in males versus females, both in mice and in humans, but that's a different story. I feel like you're doing really well. I hope I'm not overwhelming you. I think, but let's pause for a second. Do we need to stand up? I think we've got it. Like conceptually, if I can just leave you with that, I'm happy, but what questions do you have, or how are you doing with all this? Any comments? Is it good? Should we keep going? No. Elena, any comments? So the telomere, the short telomere, let's say, in aggregate, is a really important paradigm, I think, relative to DNA repair. One, because we all get it, and two, because there's different mechanisms of how disease happens. In slow turnover tissues, we talked about these second hits, so nicely articulated. In high turnover tissues, it's complete attrition of a stem cell that is replicating a lot and it's gone. So that's, and these processes happen with aging in all of us. So this is a particularly relevant mechanism of DNA repair or lack thereof. So it turns out, I'll just show this academically, but we talked about immune deficiency being a canonical phenotype of double-strand break repair syndrome. So it happens in the telomere, these other strange ones here. But it happens to be, in retrospect, that all DNA double-strand break syndromes pretty much have lung disease. This is because they haven't described it yet, but I'm sure that about half. So the lung and slow turnover tissues, also the brain, are susceptible to DNA damage. And because this is a very gentle mechanism, it happens to affect many, many people, and it affects adults very commonly. In fact, you might know someone who has a mutation in telomerase and lung disease because it's so common. Okay. Should we talk about the long telomere for a second? So why is it that the short telomere is so programmed, right? And maybe I'll finish off with this idea. So why is it that we are programmed to go downhill, as Elena exclaimed or emphasized? Right? So why is it that organisms regulate the amount of telomerase there is? So there's such a disparity, 92 to 10, that it ensures that we're going to age.
I guess too much could cause cancer.
Yes. So that's exactly right. And so people with, so I'll use this as an example, because this is a very nice experiment done by David Felter, who was a human genetics student in the Grider lab. And they bred an oncogene with short telomere mice and long telomere mice. And guess who won? The short telomere mouse won. So it's not downhill. Perhaps you and I can draft a thank you note to the short telomere. Like what seems to be canonically a downhill phenomenon, a limiting phenomenon, and here the long telomere mouse dies more often from an oncogene. That's interesting, right? So then you may have heard about our story. I don't want to talk too much about our work, but I will mention it because I think it is a parable of sorts that exemplifies the point you just raised. Thank you so much. So, sorry, I'm going to skip here, but it's these people with super long telomeres that you want to know what if anything is wrong with them. Do you guys know this story already, or have you heard of it? You've heard of it a little bit? Yeah? You know about it, or? No, okay. So at the risk of overlapping with some of you, so, all right, Emily was just graduated from our program too. So these are people who have mutations in, I'll tell you, so not only is the amount of telomeres, if you've read the review that I sent Cody that I wrote, whenever I write these reviews, I talk about all the suppressors of telomerase activity. So there's amount, right? And this repression is related to the promoter of TERT, which has very unique sequences, but cancers acquire mutations in the TERT promoter to increase its activity. So you'll hear about that. But telomerase is there's not just little of it, but there's also proteins here, many of them, that negatively regulate telomerase elongation. There are so many safeguards on telomerase elongation to prevent excessive elongation. It's highly regulated. So don't believe anybody who calls you and says, please invest in my company. I'm trying to reverse aging by elongating the telomere. I hope I will convince you that will never work, because it's not one safeguard, it's like 10 safeguards. One of the safeguards are mutations in this gene called POP1. And these families have mutations in POP1. And this POP1 gene, for reasons we don't understand mechanistically, prevents elongation from telomerase. So if you're missing one copy of telomerase, you get these excessively long telomere lengths. And these people have all sorts of malignancies. They get brain tumors, they get kidney cancer, they get thyroid cancer, they get melanocytes and melanoma. And this is a mechanism of tumor predisposition that is not previously known. Like, it's not a tumor suppressor, it's not an oncogene, it's just lifespan. If you have a good name for it, let me know. It's a youthfulness mechanism. The people in this, the ones that have the POP1 mutations, they look younger. They don't go gray. It's exactly the opposite of the other people. But guess what they get in their lung? They get lung cancer, too.
One of the students asked a question in the
Yes,
chat.
please, yeah.
They said, is the lung severely impacted because cells in the tissue are mostly in G1 phase, so an athg machinery is available instead of HDR machinery?
Yes. The NHEJ machinery is error prone and can't keep up in the setting of excessive damage from both the telomere and the genomic damage.
Can I also ask a question?
Yes, please.
So with the POP1 story, what's interesting, and this is like a very weird question, but there are other organisms like lobsters that have like very active telomerase, from my understanding. So what's the balance? Like, because they say that this idea that they like live for so long without getting these types of malignancies. So what's like the balance?
How long do lobsters live?
I don't know. There's like this idea that they live forever.
Yeah.
No one actually knows. Like there's no determined lifespan for lobster. I can't find one.
Would a better example be like those turtles that live for like 200 years, or those tortoises or whatever?
Yeah.
They say they've never observed a lobster to die from old age. It's usually been exhaustion, disease, or predation, but not from.
Well, yeah, there's also, I remember the paper.
Someone needs to capture one. That's easy.
The shark has been around for like
Oh, it's a Greenland shark for like 200 years or something like that.
No, maybe like a lot, yeah, no.
I would love to learn more about that. I feel inadequate when I don't know. The only thing I know is that elephants, they solve this problem by having multiple copies of P53. So they are hyperploid, I think. This was only Kirby will remember that paper, maybe. But not so long ago, Nature Genetics, I can send it to you, but I think long-lived organisms find different solutions. I know the human limits the range of telomere length, that's for sure. So, were you gonna say something? Yeah, these patients get all sorts of cancers, and in honor of the immune deficiency, that these patients get the opposite of immune deficiency. They get a lot of lymphoma. So it's almost the perfect mirror image of the immune deficiency. In fact, if you sequence their blood has more mutations than they should for their age, right? So I think I'm going to go to that slide and finish there. That this is an experiment where you take the blood from a long telomere patient and you create single cell colonies of hematopoietic progenitors, and then you sequence that, and then you recreate the phylogenetic relationships. Okay, so let's walk this, and then I'll finish here. In the top person, this is a healthy control, who's the uncle of this person, and this person has a FOT1 mutation, this one doesn't. You see that each of the lineages here, this is the number of somatic mutations. They have what, at 68, about an average 400 somatic mutation per genome. But here, they have about 700. Oh my gosh, look, but this person is younger. Oh no, it's paradoxical. The younger person has premature aging because their cells live longer. So what do we learn from this? I think just for the sake of, interest and not leaving this topic sort of hanging, we have many genes. There are 4 other genes that cause the same phenotype, and our lab is very interested in identifying multiple genes that excessively lengthen the telomeres, and I think that in aggregate, this cancer predisposition mechanism will account for as many cancers as BRCA2, for example. So it's a lot. So how should we conclude? These disorders, as these disorders in the color scheme before, are all autosomal dominant. They're all haploinsufficient. They affect, I guess, not DNA, they kind of affect the DNA because the telomere length is the primary driver of the disease. But it is very tightly regulated, much more exquisitely regulated in the human at least, with the lobster and elephants and turtles aside, that it's an exception to the DNA repair rules of autosomal recessive that we talked about in the beginning. You can get away with probably, maybe if someone works as a Navy SEAL and they have only one copy of XP, their risk in light of spending so much time in the sun is going to be higher for melanoma than someone who's XP wild type. But in normal conditions, whatever those are, that person probably has enough repair capacity to handle the damage. What else should I say? these pathways are highly perturbed. They're important for life on Earth, life in the setting of the sun. How organisms that live in very adverse conditions deal with extreme, like we know that bacteria can live in acid, highly extreme acidic conditions. How do they manage that is really a curious thing. Let us not forget about studying fundamental things as we restructure our entire research system. Let us remember that we can learn things from model organisms that will help us understand human disease. And I hope I've convinced you that the study of human disease during using these complementation groups, even in rare disorders, has given us insight into understanding very common conditions like BRCA1 deficiency. So I feel like I should stop here. I can talk more, but I think we covered a lot of ground. What do you think, Elena?
Good.
Yeah. What are the questions do people have? Yes, please.
I have a question about, I guess, like how telomere length is first, I guess, decided when you're like an embryo. Is it like based off your parents' telomere length? Like, do the people with really long telomeres, do their parents also have long telomeres, or is that like in the early cell divisions, do they get lengthened?
Great question. So I'm going to use that as an excuse to mention one more thing. So sorry. So you see here, this is a classic cross. I'll ask you guys back a question. What's the telomere length of this wild-type mouse after it has been through generations of shortening? Is it the same as a wild-type mouse that's from the Jackson lab? Or is it going to be different? Different. In which direction? Shorter. And it's going to be shorter. How long will it take for it to elongate back to the Jackson lab threshold? 5 generations. I can send you the paper. It's amazing. So even across generations, the elongation capacity is finite. And in the families that we study with autosomal disease, sometimes the offspring who are wild type not only have short telomeres, but if they smoke, they get the same lung disease as their siblings. So that's striking, right? That's like a peculiar exception to Mendelian genetics. And we see the same thing with the long telomere. Someone who's wild type who inherited the long telomere length from the parents could develop a milder spectrum of cancers. So this is why this biology is so important for genetics in general, because not only do people who have super long telomere length and POT1 mutations get cancer, the people on the boundary of the normal nomogram also get cancer. And this is, if you're ever bored, if you're ever bored, look at the gene losses for cancer. All of those people have hits near POT1 and TERT that elongate the telomere. So this is a fundamental mechanism of cancer predisposition. I can say more about that, but that gives you a sense that our parental telomere length influences our telomere length independent of the genotype, right? Those wild-type mice, they have the same exact genome as the parents, but the only thing that's different is the telomere length. They call it, some people have called it the telotype. It's a genotype of sorts. Kirby, you were going to say something?
Okay. I was going to ask actually about GWAS, like variants that have been identified associated with telomere length. Are there any that have been identified associated with longer telomere length, but without malignancies?
Oh, that's a great question. If anyone wants to study GWASs, this is a classic example of interpretation. So when people first did the GWASs, so, right, it goes 1 to 22, and this is the p-value, right? The most common hits was on at TERT in chromosome 5. And if you read the early Nature Genetics papers, I'm like, we found TERT as the most common associated SNP in lung cancer. It must be the short telomere because short telomeres cause aging and aging is cancer related. But it turned out that all those assumptions were false. You know, I wrote a review on this, that just because you have the association with a SNP doesn't give you the direction. Is it protective? Does it increase risk? So all those SNPs associated with cancer, if you look carefully enough, now that we have the telomere length data from whole genome sequence, is associated with long telomere length. So that's an interesting lesson in interpretation of GWAS from our fields. Yeah, great question.
So I'll just make this, I've always thought that we had all this DNA repair mechanism because DNA polymerase is such a sloppy enzyme. But in fact, I think we have this DNA polymerase has to be a sloppy enzyme because of the repair mechanism. Because if the repair mechanism could correct all of the mistakes that DNA polymerase makes, we couldn't have evolution.
That's right.
So there has to be a balance between the two, and polymerase is never going to get better.
That's right. But it's remarkable that different pathways have evolved to fix the mishaps of DNA polymerase. So mismatch repair, for example, it's like you have one base, that's the parent allele, and the other one doesn't match. DNA polymerase put it there by mistake, and then you have the entire mismatch repair deficiency pathway that evolved to fix that, or base excision repair. So there must have been a reason why these evolved, so, because they're very intricate.
Right, so I would think that the repair mechanisms evolved to correct environmental insults, but then DNA polymerase had to get worse because the repair mechanisms were so good.
Yes, or exogenous, right? We talked about you said oxidation in the beginning. I don't have a good sense because it's hard to quantify these things, as you see, because cells that are damaged and are short-lived, as Elena said in the beginning, if you have blood cells that live for 6 hours and they're damaged, they're going to leave. That's not going to matter. It's the long-lived cells that we live with long-term. So how much, yes.
I think Dr. Ravi also mentioned this about what benefits about the balance between accuracy and evolution, basically. What's good for the species may not be good for the individual. He was saying, because you need some variation for evolution, but then the same mechanism also can compare to the life survival of one individual, even though it's beneficial.
I think another observation would be a very good project would be to study the telomere lengthening or shortening in lobsters. Because there you could do all of your mistakes.
I think the issues of aging and longevity in non-human, non-vertebrate models is wonderful.
Studying sharks, so much more sooner.
Maybe non-rodent, non-human models is great. Like if anybody's bored, I think we can do a literature review, have a party. We can dress up as those animals. We can figure out who has which spirit animal. Zoe's spirit animal is the turtle. Yeah. In case you didn't know. Okay, before we go, I just wanted to thank you so much for your help with this and participation. I think these times are hard, and I know it's a delicate moment for people who are starting their career, but you're well positioned, and I appreciate all that you did today and your participation. And you came up with a lot of insights that I think are not so obvious in the literature, so I appreciate that.
This was wonderful. Thank you for coming.
Thanks. Take care. And thanks to the people online.
I know, right?
It's nice, Elena. I must have called Elena 6 times about this class. I
I don't have them with me right now.
I didn't grab it.
I have them in my key drawer at home, like, so
At
I'll
home?
forget, yes.
Oh my God, I
Because
really love that.
you need the, oh, you don't have to put it here.
It's okay, I'll take it tomorrow.